Right, thank you. I'm Betsy Luczak, an assistant professor in the Division of Cardiology, and today I'm excited to share with you a compelling opportunity in cardiovascular medicine, our technology that aims to transform the treatment of atrial fibrillation through the development of novel drug compounds. First, let's discuss a significant medical challenge, atrial fibrillation, the most common cardiac arrhythmia and a leading cause of death due to the increased risk of heart failure and stroke. AFib occurs when the electrical signals in the upper chambers of the heart become chaotic, resulting in an irregular heartbeat that is often faster than normal. About 6 million Americans suffer from AFib today, with these numbers expected to double by the end of this decade. Unfortunately, there is no cure for AFib. Current therapies, such as medications and catheter-based ablation procedures, often fall short, leaving patients with intolerable side effects and recurrent AFib, often needing additional therapies and repeat procedures. But what if we could offer patients a better solution by creating a more targeted therapy? Decades of research from our lab and others have identified the enzyme CaMKAR as a key driver of cardiac arrhythmias, including AFib. CaMKAR is activated by calcium and is an important signaling molecule in excitable cells, including heart muscle cells, for heart rate regulation. CaMKAR sits at a critical intersection, making its pathological activation cause dysregulation of the heart's electrical stability, causing irregular heartbeats. Genetic expression of highly specific CaMKAR inhibitor peptides have illustrated the benefit of CaMKAR inhibition as a therapeutic. In our lab, we demonstrated that expression of this CaMKAR inhibitor peptide in heart muscle cells significantly reduces the induction of atrial fibrillation in a mouse model of acquired AFib. Currently, no CaMKAR inhibitors have reached clinical approval. Several companies have attempted to bring a small molecule CaMKAR inhibitor to the clinic, and many of these have been abandoned due to adverse effects or low potency in animal models or patients. Recently, a small molecule inhibitor has made it to phase 2 clinical trials. However, this molecule was likely developed using the same traditional cell-free assays that have previously failed. The biggest challenge now is the discovery of novel CaMKAR inhibitors, which has been limited by the lack of adequate screening tools. Our solution addresses this challenge directly. We have developed a new tool called the CaMKAR Activity Reporter, or CaMKAR. This genetically encoded biosensor allows us to measure CaMKAR by detecting changes in the fluorescence of GFP. This enables real-time monitoring of CaMKAR activity in living cells. In our recent publication in Science Translational Medicine, we fully characterized this biosensor and demonstrate its enhanced sensitivity, selectivity, and fast kinetics, making it suitable for live cell assays at high throughput. Using our CaMKAR biosensor, we developed a comprehensive drug discovery platform and conducted a proof of concept screen. In this screen, we used cells expressing the CaMKAR biosensor and treated them with a library of nearly 5,000 compounds that are approved for human use. We discovered 5 drugs that were previously unknown to be CaMKAR inhibitors. The best of these inhibitors, ruxolitinib, was able to significantly reduce atrial fibrillation in our mouse model of acquired AFib. Our platform is unlike any other discovery method in use today. It is fast, specific, and relevant, enabling us to screen compounds in live cells. While ruxolitinib is a CaMKAI inhibitor, its primary mechanism as a JAK inhibitor make it unsuitable for use long-term as an antiarrhythmic. This highlights the need for the development of novel CaMKAI inhibitors that are developed specifically for cardiac applications. Our goal is to innovate arrhythmia therapy by developing a new class of drugs designed to inhibit CaMKAI. Our strategy pairs our CaMKAR-based screening platform with computational design, develop new chemical structures that are predicted to inhibit CaMKAI. We are using both cell-free kinase activity assays and CaMKAR-based biosensor assays in cardiac cells to find inhibitors that both directly inhibit CaMKAI and also are functional in the heart. With funding from 2 Johns Hopkins-based translational research awards, we've been able to begin this discovery process. Using ruxolitinib as a scaffold, we used computational design to develop new inhibitors that are predicted to inhibit CaMKAI. Using cell-free kinase activity assays, we determined the IC50 of our design compounds for both CaMKAI and JAK. Our goal in this initial round was to eliminate or reduce JAK inhibition. All of our compounds improved selectivity for CaMKAI compared to ruxolitinib by reducing JAK inhibition while maintaining CaMKAI inhibition. Next, we wanted to improve the potency of our inhibitors for CaMKAI. We did this by expanding our library of CaMKAI inhibitors. Using our CaMKAR-based platform, we screened 24,000 kinase-targeted compounds. Over 22,000 passed our initial statistical validation, and 6 of the 38 compounds we have tested so far directly inhibit CaMKAI, giving us additional scaffolds to use in our computational design. We also validated these compounds, both design compounds and screen hits, in our cardiac myocyte assay. Here we express the CaMKAR biosensor in isolated rat myocytes. And we activate CaMKAI signaling using electrical field pacing. Here I'm highlighting 3 compounds that had similar IC50 in our in vitro assays. However, only 2 of these compounds significantly inhibit CaMKAI activity in cardiac cells. This really highlights the advantage of our platform in finding compounds that are functional in the heart. So far, our work has identified 2 novel chemical structures to use as scaffold for our future computational design. Our goal in the next round is to improve the potency for these compounds for inhibiting CaMKAI. We have a tremendous opportunity to treat atrial fibrillation for millions of patients. Our approach uses CaMKAI as a validated target and mediator of cardiac arrhythmia. What sets us apart is our CaMKAR-based biosensor platform, which we have validated through proof-of-concept screening, including the successful identification of ruxolitinib as a CaMKAI inhibitor. We've already screened 24,000 compounds and utilized computational design to identify promising new CaMKAI inhibitors. We are now synthesizing our second round of design compounds to test in our models. Together, our founding team began this work at Johns Hopkins. The work is continuing to go on in my lab, and I have established key collaborations within the Johns Hopkins School of Medicine. Oscar Reyes-Gallido was instrumental in developing our biosensor and the discovery platform. And Mark Anderson is a world-renowned expert in cardiac CaMKAI biology and has industry experience in developing CaMKAI inhibitors. We are now seeking seed investment to complete lead discovery and optimization. positioning us to establish a new company to advance a CaMKAI inhibitor to clinical development to meet this critical unmet need in cardiovascular medicine. Thank you for your attention, and I welcome any questions.